two wonderful PhD students, Iris Bennett, who's a research scientist at Corteva AgriScience in Iowa, and also Tuhim Majumder, who's a postdoc researcher in Duke Biostat. So the outline is pretty simple. After introducing the problem itself and the data model that we'll use, I'll talk about fitting the model, and then to go back to the application, which is to classifying viruses. So there've been over 700 million SARS-CoV-2 or COVID-19 cases worldwide, with nearly 7 million fatal cases. And I won't go into the symptoms. Too many people know about the symptoms of COVID. Most patients exhibit mild to moderate symptoms, with the fatality rate being much lower than the 10% fatality rate for the 8,000 reported cases of SARS-CoV-1 for the SARS-CoV-1 outbreak in China. MERS-CoV, or the Middle Eastern Respiratory Coronavirus, affected the Arabian Peninsula in 2012 with a fatality rate of about 35% for the 2,500 cases. That's crazy. It's been speculated that a lot of the milder cases were not reported, because 35% is just like, wow. And there have been other viruses that have symptoms similar to coronavirus, examples including dengue, which is prevalent in the summer and fall season in India, and hepatitis B. A false diagnosis can be fatal, and thus accurate testing procedures are important. In the rapid antigen test to detect coronavirus, a positive result is reached if the sample contains the DNA signature of the pathogen. And collected samples, if fully available, are almost equal to the reference genome for the viruses, so for when the virus is present. And that makes prediction easy. But in practice, there are many occasions when only part of the full DNA sequence is available, and reasons include comorbidity, when there's like a mixture of deadly viruses or deadly diseases, so that there's a corruption in the pathogens mix and mutate, or technical errors in sequencing, or that lead to gaps or corrected segments. So the large part of the DNA sequence needs to be trimmed. So under that scenario, classification of viruses can be challenging, and that's the situation that we'll be talking about here today. We'll also be considering the efficacy of using higher order Markov models, and specifically sparse Markov models. So that's kind of my interest, truthfully, for the modeling and classification task that we examine. And the partial DNA sequences that we'll be using are from the 4 viruses that we mentioned before: SARS-CoV-2, MERS, dengue, and hepatitis B. And we'll begin with an introduction to sparse Markov models. So we're in a situation where we have a categorical time series taking values in some finite set, which we'll call sigma. And most are probably familiar with higher order Markov models. The assumption is that conditioning on just the last M state for some finite M is sufficient. More formally, the conditional distribution of what happens right now, given all of the past, is equal to the conditional distribution given only the last M state. So given the last M states, I have all the information that I need. And of course, when I have an mth order Markov model, it's associated with a first order Markov model if I take my states to be M tuples. There are drawbacks associated with higher order Markov models. The big problem is that the number of conditioning histories, the number of M tuples, is exponential in the order M. And so what happens, because there's so many transition probabilities to estimate, what can happen is that people end up using lower order models when a higher order model would actually be better. Another drawback is that there's a lack of flexibility in modeling. For example, if I have size of sigma to be 4, so if I have ACGT as my state space, and the third order model then would have, 164 histories, so 192 transition probabilities to estimate. But if I wanted a fourth order model, I'd have to have 768 transition probabilities to estimate. There's no in between, basically, is the idea. So there's a lack of flexibility, basically, in modeling. Sparse Markov models kind of help with both of these problems. So of course, I haven't told you what the model is yet. So a sparse Markov model of order M, or an mth order model, is one where M tuple histories are clustered into groups that have the exact same conditional probability distributions. The clustered M tuples, so we're writing here that there are n different clusters along with the conditional probability distributions associated with each cluster, give the sparse Markov model. So instead of having size of sigma to the power m times size sigma minus 1 transition probabilities, we've replaced that size sigma to the m by eta. So this gives an opportunity to have many less parameters in our model, and the model itself helps with navigating the variance bias trade-off. Bias from having a model that's of order too low and doesn't really fit well, but variance from having too many transition probability parameters to estimate. These sparse Markov models have been considered under various names, and the interesting thing is that they have only been talked about since 2010. So there's a lot of area for work in sparse Markov models, whereas Markov models have been dealt with for over 100 years, right? And okay, so, but different names in the literature, if you're actually looking for minimal Markov models, partition Markov models, sparse Markov chains are some of the names that have been used for them. And that's actually one of the excitement of sparse Markov models for me, is that there's a lot of area for research and for developing algorithms related to them. But actually, in truth, we see from the beginning of this slide that they are actually a special case of higher order Markov models, of an mth order Markov model, where we've done this clustering. So, of course, I can assume any kind of model, but I need some rationalization. It needs to be fit to data. So the data kind of needs to tell me what model should be fit. So fitting the sparse Markov model is then a clustering problem. I'm clustering, instead of clustering data points, as we typically think about it, I'm clustering empirical conditional probability distributions is what's happening. So what these determined by the clustering is how these gammas are determined, how the model is actually determined. So it's a clustering problem, and so a lot of work has been done on clustering, and we can draw then on that work. It's basically the same problem, and that's actually what has been done in this work here. So we need to determine the number of groups. We need to determine the M tuples in the groups themselves. So that's what the model fitting problem is. It's a clustering problem. So what we want to do is we want to calculate empirical conditional probability distributions, right? Just using counts from the data. And we want to cluster these distributions when they are close in some sense. So again, we're clustering not points, but we're clustering distributions, conditional probability distributions, vectors of length, size of sigma. So for AGT-CT, size of sigma would be 4. So that's in our application. So we've already said there's been a little work on fitting sparse, or on Markov models themselves, and little work on fitting sparse Markov models. Garcia and Gonzalez-Lopez in 2010 and 2017, using an agglomerative hierarchical clustering approach in which two clusters of histories are combined when their combination reduces the BIC of the fitted model. And in 2014, Jaskinen and that research group set a uniform prior over possible partitions of histories and use agglomerative hierarchical clustering at each step so that the posterior probability of the partition is maximized. And then basically the same research group, just with the name switched in 2016, made the algorithm faster or more efficient. So that's kind of the situation that we faced. Not a whole lot of work on the model fitting. The work that had been done didn't seem to be all that effective. So Dr. Lahiri and I kind of turned two PhD students at different times loose on the problem. And we'll talk just a little bit about the first approach. We'll kind of focus on the second. So the first approach was Iris Bennett, and the work is published in Computational Statistics. Iris used what she called a Gibbs sampler Dirichlet process mixture model to do the clustering. She drew on the literature on topic models in her work. And so of course it's Bayesian. She said Dirichlet process priors to multinomial distributions, and thus ended up being able to use a Gibbs sampler. So just to give you a brief overview of her algorithm, after setting an initial partition of histories, each history is removed from its current cluster and placed in a new one that maximizes the posterior likelihood of observed data. The process is repeated many times with counts after some burn-in period kept for the partition that gives the highest posterior likelihood. And in the end, the maximum posterior value is chosen as the clustering. What helps the algorithm to work is that while there are many, many possible partitions, in fact, the Bell number is the number of possible partitions, the Gibbs sampler never actually reaches partitions with low posterior probabilities. So that's kind of what helps it to hone in on the right answer. So here's the other approach. It's by Tuhin Majumdar. It's basically his work. So Tuhin, on the other hand, used a regularization model or penalized functions to fit the model. So this probably looks pretty familiar to a lot of you. So what we want to do, the POJs here are empirical conditional probability distributions. The B's are centroids. It turns out that they are probability distributions as well, and that can be shown. So we want to then minimize this function. We have lambda is a parameter that kind of drives the regularization and also the weights. So we're penalizing so that we can kind of coerce things to combine is the idea here. So again, lambda is the penalty parameter. I just made things look crazy. Maybe, do I want to escape? What'd you say, dude?
Just to click somewhere else. I think you just selected the whole slide.
Oh, that's what I'm trying to do, but
It didn't work. That's interesting.
Not that bad. No, okay, it's working again now.
Go back again.
Yeah, this is where, okay, thank you. Thank you so much. Okay, so this is the approach. And so one penalizes the distance between different pairs of probability vectors in order to identify the identical ones. I've already said these centroids, these B vectors, turn out to be probability distributions themselves. So the good thing is that the number of groups, after we finish this clustering problem, the number of groups eta and the groups themselves will be evident. So we want to solve this convex clustering problem. There have been several efficient algorithms to solve it. The one that we're honing in on for reasons that are probably obvious is the algorithm of Eric Chi and Kenneth Lange, published in JCGS in 2015. They gave an elegant method using augmented Lagrangian methods. We'll continue to use the notation of our problem, but we're going to note as we go throughout, we're going to talk about basically the work of Chi and Lange, because that's what we use to do our problem. And we note that in their paper, they laid out the problem and its solution in a much more complete manner than what we're discussing here. So we put the 2 norm, for example, on the difference between the vectors, but they worked the problem in the more general setting of an arbitrary norm with the requirement that the proximal map for the norm be readily computable, or equivalently, that the projection onto the unit ball of the dual norm should be straightforward. So I advise you to read their paper for details. It's a wonderful paper. Yes, well.
This pi hat is also as a probability vector as a central usually the solution is a probability vector.
It turns out that the solutions are a probability vector, and Tuhin showed it. Yeah. With that, which was kind of it was not an obvious result, but they are. Yes, that's a good question. Okay, so in Chi and Lange's work, they discussed 2 solutions to this convex clustering problem, one being alternating direction method of multipliers, or ADMM, the other being alternating minimization, the alternating minimization algorithm, or AMA. But both use or both begin with this constrained optimization problem where splitting variable V has been introduced to capture the difference between these centroids. And the script epsilon is the set of distinct edges associated with, well, the non-zero weights, basically. Chi and Lange noted that in both of these methods, the ADMM and the AMA, one incorporates an augmented Lagrangian in this way, where B, V, and psi are matrices, and little b, little v, little psi are columns in those matrices respectively. The splitting variables allow one to update B, V, and psi sequentially, given all the other variables. Chi and Lange showed, and this is important, that AMA is much faster than ADMM, especially when the weights are sparse. And also another advantage is that in using AMA is that V and psi need to be updated at each step, but not the V. So following their suggestions, we used AMA in our work. So we need to update B. So here we've represented B at time t and psi at time t for the estimates at the t step, and then the update to time t+1 is handled in this manner, where G is specified as here. And let's see, this P is a projection. So it denotes the projection of this quantity onto this set, the set here. And the convergence criterion uses the dual problem and duality gap, and of course, is discussed in more detail in Chi and Lange's paper. As far as the choice of weights, Chi and Lange discussed how the weight matrix can dramatically affect the quality of the clustering or the clustering path. They recommended using K nearest neighbor based weights. So the weights that they recommended are as represented here, where this L superscript K and subscript IJ, that's the indicator function that pi I hat is one of the K nearest neighbors of pi J hat. And this other part, this Gaussian kernel, is to further encourage or actually further discourage vectors that are far apart from coalescing. This phi is some non-negative parameter. Actually, phi equals 0. If we took phi equals 0, we would get uniform weights, all of them 1, 1 or 0, due to the indicator function. And as noted earlier, limiting positive weights to nearest neighbors improves both the computational efficiency and the quality of the clustering. An important thing is, and I did it again, oh, okay, I did it. Except for it took me right. It doesn't like this slide, so I'll get past this slide. So lambda equals 0 would correspond to a single cluster, right? Let's see. Let's go back here. So if we had no penalization, then we would just want to set all the b's equal to the pi's, and so we would just have this size sigma to the m singleton clusters. But as we increase the lambda, then the coalescing starts, and the higher the lambda, then in the end, if we just kept increasing lambda, we could end up with just one cluster. So the good thing is in the solution path, we get to see all the different possibilities. And so that's one wonderful thing about this approach to convex clustering. The other is that because it's a relaxation, so we have convexity so that we don't get stuck in local minima. So that's a good thing. So we used exactly lambda, we used exactly this Chi and Lange method. The one thing that seemed to be kind of missing was how in the end do you actually choose which lambda you want, which clustering do you want? So it gives us a whole path. But Tuheen, this is Tuheen's work, truthfully, Tuheen decided to choose this tuning parameter lambda and thus the clustering assignment to minimize BIC over a grid of lambda values. So the BIC defined here, this L of lambda and this eta of lambda are actually the values that come from this a specific lambda. So they are a function of specific lambdas, but then we compute this as at different lambdas and then choose the one that's where BIC is minimized as our optimal cluster assignment. And Tuheen gave theoretical results that indicate that for a range of lambda values, one can perfectly recover the true clusters for large N. And now we'll talk a bit about the algorithm to classify the virus or applying it to that specific problem. So 4 different viruses, the COVID-19, MERS, Dengue, and hepatitis B. There were samples from 500 individuals drawn from the NCBI database, 200 from COVID-19, 50 from MERS, 100 from Dengue, 150 from hepatitis B. The MERS samples were collected from Arab countries in 2012. Dengue and hepatitis B samples were collected from across the world over the last 30 years. And the samples from COVID-19 were selected at 4 different strategically placed time points to kind of try to catch different strains of the disease. The NCBI database contains reference genome sequences for the viruses, representing their ideal genome structure. And as we said earlier, when full sequences are available, little classification is needed because the sequences are nearly, or the data that we have, the data is nearly equal to the reference sequence. However, in practice, it could be that we don't have full sequences, so we dealt with that scenario or tried to deal with that scenario. So this is the procedure. First, a reference sparse Markov model was built from the reference sequence for each virus. So we estimated a sparse Markov model for each reference genome sequence. And then next, randomly selected contiguous segments of the genome sequence for each sample and used that to compute the likelihood of that segment under each of the 4 reference models. And the model with the highest likelihood is the one that was chosen in the end for the model. Just to give you an idea of the data lengths, for COVID, the length was close to 30,000. For MERS, it was about 30,000. For Dengue, around 10,000, but only 3,500 data points for hepatitis B. And you'll see that that actually comes into play later. For SARS and MERS, a sparse Markov model of order M equals 4 was fit, while for the other 2 viruses, a model of order M equals 3 was fit. The longer reference sequences were associated with the higher order Markov models, M equals 4. And of course, there's a biological significance in using M at least 3, because 3 consecutive DNA codons or DNA bases form a codon, which translates genetic code into a sequence of amino acids. So it's reasonable to assume that sparse Markov models of order 3 or more will be able to explain the structure of viruses. So that's why 3 or 4 was chosen. So this table is interesting for me because I wanted kind of just to look at how the sparse Markov models helped with the modeling of data and specifically with coming up with a parsimonious model for data that had a lot of parameters. So these are the results. So with COVID-19 and MERS, there were actually, 4 to the 4. So 256 different histories that we started with. But you see in the end, the ETAs that were chosen were either 28 or 30. So that's a big reduction in number of parameters. And you see the cluster sizes here, 155, 50, et cetera. This should add up to 256 and 28 clusters in total. Here again to 256, 30 clusters in total. For Dengue and hepatitis B, there would be 64, 4 to the 3, 64 different histories. And the number of clusters ended up being 7 and 14. So that's a great reduction, but still accurately modeling the data. So that was a good thing to see. So for this each sample, what Tuheen did was randomly choose segments whose length was 100 times some epsilon % of the original sequence length, and then compute likelihoods for the 4 models to classify them to the most likely virus class. So the epsilons that were used were 5%, 10%, and 25% of the data. So he tried to look at small segments of the data and see how well the classification worked. So we'll look at 4x4 confusion matrices on the next 3 slides for those 3 cases. Here for epsilon equals 0.05, so only 5% of the data was used. A continuous segment of 5% of the data was used for the classification. And what we have down the left column, what we have here are the actual viruses, and here we have how to classify the fitted viruses. And we see that MERS and Dang actually were fit perfectly, 50 out of 50 and 100 out of 100. There was a big problem with hepatitis B, which kind of made some sense because there we had much less data than in the other cases, right? There were 3,000 in some cases of hepatitis B and close to 30,000, for example, of MERS and COVID-19. And COVID fit fairly well. But here the misclassification rate was 22.8%, which isn't so great. So didn't work so well for 5%. This is for 10% of the data used. Still, hepatitis B is bad. Only got the right answer 81 times out of the 150. COVID, so MERS and Dang, perfect again, 50 out of 50, 100 out of 100. COVID did a bit better. But for 25% of the data, things look, so here, I'm sorry, going back, 15% misclassification rate at 10%. But for 25%, the misclassification rate was only 3.2%. So that was a bit encouraging that with 25% of the data that these viruses were classified correctly in about 97% of the cases. Perfectly again for MERS and Dang. Looks like 97% for COVID and a lot better for hepatitis B. So things will start looking up, 140 out of 150. So we noted here, so this is basically what was noted at the bottom of the slide. So kind of in conclusion, as we said, epsilon equals 0.05, epsilon equals 0.10. The classification didn't work so well, but we were encouraged that with 25%, only 25% of the sequences, we could correctly identify the true viruses in about 97% of the cases. The sparse Markov model method then utilizes information from the reference genome sequence in a compact manner so that it captures the diversity of structure from different parts of the samples. And I want to thank you for your attention, and I'm just happy to be here. So any questions? Yes, Jason.
Thank you. That's a great talk. So I love how, you said that because of the sort of mechanism of your application, you had a nice way to choose the order of the Markov chain you want. And of course, you've also talked about how you can choose the number of clips using DIC, right? You can kind of tune that to make sure you. So my question is, in general, like, is there a nice methodological way to also choose the order of the Markov chain, or like, how do you do that?
That is a good question. Anderson and Goodman did work way back in 1965 on choosing an order of a Markov chain. So that's very relevant here. We just kind of, we knew that we needed it to be at least 3. But we didn't want to make it too big because we were talking about M tuples, and we didn't want to have it just to be too big. So that's why we chose 3 or 4. But so you can kind of use your intuition based on a specific problem. But there is theory, like I said, Anderson and Goodman back in 65 on starting with a first order or independent trials and then testing to see whether you should go to first order, second order, third order, et cetera. So yeah. Yeah, yes.
So I think I missed it, but when you classify those like short sequences, like when you're sampling like 100 times epsilon, like the shorter sequences, and then just checking how accurately you can classify them. Does your model like automatically classify them, or are you like training a different model to classify into the clusters? If that makes sense.
So we fit these reference genomes and we're going to compute likelihoods under the four. So for each segment, we're going to compute a likelihood under those four reference from under those four situations, those four reference sparse Markov models, I should say. And so then the one with the highest likelihood is the choice. That's all. It's totally, everything is totally data driven. Everything.